My name is Eli Merriam. I'm a member of the LBC. I run a small group that is called the Computational Neuroimaging and Perception team. And today's talk is going to be about understanding visual processing with fMRI. And this talk is geared towards beginners. So I specifically thought summer student or to people who don't do vision for a living. So I'm going to start at the beginning. It's informal, so I encourage everyone to interrupt me, ask questions. There aren't that many people here, so everyone should feel comfortable talking. Don't be shy. When Peter asked me to give this talk, he told me to focus on what we've learned about the visual system from fMRI and what we don't know, to focus on the questions for the future. And in thinking about how to organize this talk, I remembered a paper that I read when I was in the beginning of my undergraduate training that actually kind of inspired me to enter the field. And this is a review paper by Susan Courtney and Leslie Ungerleider, published in '97, so 20 years ago. And the title is, What Has fMRI Taught Us About Human Vision? And at this point, this fMRI was just a few years old, so this is the beginning of fMRI. And it was already appropriate at that moment to take a step back and ask, what have we learned about the human visual system from fMRI? So in preparing this talk, I reread this paper, and I was surprised at how current it actually seemed. A lot of the topics that they discussed in '97 are actually still very relevant today, just we filled in many of the blanks. So this talk is organized around this paper. I've just taken the topic headings, and I'm going to give a brief history of where the field has come in the last 20 years and what the remaining questions are. So this is an outline for the talk. I'm going to start with a brief justification for why we're studying the human visual system. And then I'm going to talk about retinotopic mapping, which is a way of defining the locations and extent of visual areas in the human brain. I'm going to talk about functional specialization. So each visual area does something special. And I'll talk a bit about the representation of orientation, motion, color, and object features, such as objects, faces, and letters. I'm actually not going to talk about that. Sorry, I cut that from the talk at the last minute. And then lastly, I'm going to talk about bottom-up hierarchical processing and top-down influences on visual cortical areas. Now, these last 2 points are enormous. I mean, there's no way that a 15-minute tutorial talk could cover them. But I want to give you a flavor of the sorts of questions people are asking and what I think the future is going to hold for addressing those questions. Okay, so why study human vision? The visual system you can think of as kind of a machine. So I take an engineering approach to studying vision. We can characterize the inputs very well. We think a lot of the stages of visual processing can be characterized using tricks from engineering. And a lot of the neural processes that support those computations can be interrogated very directly using invasive physiological techniques such as single unit recording or calcium imaging. So given that that's my interest, a lot of people have asked why study humans with fMRI, which is a coarse technique and doesn't offer us a lot of the same insights as invasive physiological methods. And there's a very nice review paper by my collaborator, Justin Gardner, that basically defends the approach by saying, look, we want to span the levels of analyses from computational to physiological to perception. And studying the human brain is an important intermediate step in that chain. Furthermore, ultimately we care about how the human brain works because we're interested in mental disorder. And in order to cure mental disease, we have to understand the human brain, and there are important differences between humans and animals. So understanding basic physiological phenomena in humans is an important step in that process. So the short take-home message is the human brain is the best model for understanding the human brain. This is a great review, by the way. I highly recommend it. Everyone should read this. Okay, so retinotopic mapping. There are 4 criteria for defining a visual area as a visual area, summarized as fact. So each visual area has unique physiological properties, unique connectional architecture, architecture within the visual area, and topography. So that's the representation of the visual world. And while we can gain insights into all 4 of these with MRI is kind of best suited for studying topography. So what do I mean by topography? This is a schematic of V1, which is the first visual cortical area. And V1 has a map of the external world. Everyone know what I'm talking about when I talk about topography in V1? Yeah, raise your hand. So this is a little cutout part of V1, which is shown here. This is the occipital pole. And the upper vertical meridian, that's its location in space, is located in the ventral part of V1. And there's a map so that the lower vertical meridian is located in the upper bank of the calcarine sulcus in V1. So there's a map of visual space, and fMRI is capable of describing the characteristics of that map. So in a typical retinotopic mapping experiment, subjects fixate on the center of the screen, and a circular disc kind of slowly expands through visual space, or a wedge slowly rotates around visual space. And as those stimuli expand or rotate, they activate successive parts of the retinotopic map and create a traveling wave of activity. So this is an example experiment where I had subjects, actually this was developed in the mid-90s, but I do this all the time here. Subjects fixate, and this wedge slowly rotates around the fixation spot. And what this does, this is a schematic diagram of a bunch of receptive fields in V1 that might be covered by an individual fMRI voxel. So voxels are much larger than neurons. There are many neurons with different receptive fields within that voxel. Those voxels all lie at some portion of the visual space, and as the wedge rotates, it goes in and out of receptive fields, creating a kind of wave of activity. And the location of that wave is dependent on where these receptive fields are. So if the receptive fields are over here, when that wedge goes through, there's going to be a positive response. For these same receptive fields, that response will go back down to baseline when the wedge is over here. So there's a traveling wave of activity. I can then project the data from each fMRI voxel onto the cortical surface. I'll be showing you lots of pictures like this in the talk. This is the raw T1 weighted fMRI image. This is a 3D cortical reconstruction of the surface. And this is a flattened patch of V1. I can then characterize the time course of the response for each voxel in V1 using a Fourier transform. So this red curve here is actually the measured fMRI response to a voxel over here in the brain that responds when the wedge is over here. Okay? And I can characterize two parameters of the response, basically the amplitude, which is the size of the evoked response, and the timing, which is characterized by the phase. So each voxel gets an amplitude and phase that allows me to relate its time series to the position of the wedge. Make sense to everyone? So this is one voxel in V1, this is another voxel. You can see these two voxels have different phases. And then I can color code each voxel by its preferred phase. That's a retinotopic map. So when Ungerleider and Courtney wrote that review in '97, this was the state of the art. This is a science paper by Marty Serino and others from MGH showing a coarse retinotopic map in V1. V1 is located over here, and each color corresponds to a different location in the visual map. This is the angular map, and this is the eccentricity map. And from these maps, you can identify the boundaries of about 4 different visual areas. So a visual area is defined as a full representation of colors. So V1 is over here, this is lower V1, this is upper V1, then V2 goes from purple to red, then V3 goes from red to purple, and so on and so forth. So in the mid-90s, this was used to define a handful of visual areas. But there's been a lot of progress in the last 20 years. About 10 years after that, people learned how to combine data from multiple subjects, and people started scanning 3T with better surface coils, and they discovered more areas and elucidated cleaner maps. So this is a retinotopic map averaged over 15 subjects, and in addition to V1 and V2 and V3, you can now clearly see V4 and V3A and B, and these 2 new areas which don't exist in monkeys, LO1 and LO2. So progress in identifying retinotopic maps. And actually one of the interesting things about this result is that LO1 and LO2 don't exist in monkeys. So we've already learned something about the human brain that we didn't know from physiological studies in monkeys. Okay, but that's not the end. People have gone up into parietal cortex and changed the task around a little bit and discovered even more visual retinotopic areas. So beyond V3A and V3B, there's an area called V7, which some people call IPS0. That's at the base of the intraparietal sulcus. IPS1, IPS2, IPS3, IPS4. Okay, so there are a lot more visual areas. Again, many of these areas don't exist in monkeys. And one of the major open questions at this time is what these areas do. Are they specialized for particular functions? How did they evolve? So on and so forth. Lots of unanswered questions. There are also sort of unexpected maps. So people have looked for somatotopic maps that have both visual and somatotopic properties. So in this experiment from Marty Serino, also in 2006, subjects laid in the scanner and had air puffs at different locations on their face. And he discovered a region in parietal cortex that has a map of face location. In fact, you can activate these areas with visual stimuli or face puffs. They have congruent visual and somatotopic representations. There have been efforts to systematize all of the visual areas that you can record. So this is a very influential paper from Sabina Kastner's lab a few years ago, scanning 50 subjects and trying to identify by hand each individual atlas location, retinotopic location, and then averaging the ROIs from each individual subject using some probabilistic atlas tools. And what they found is that you can identify reliably 25 areas in the human brain. So this was published in 2015. There have already been more areas added. So this is not the final list of visual retinotopic areas in the brain. Another advance has been a move away from the traveling wave protocol that I told you about before. So when I explained retinotopy, I said that the stimuli were periodic and moved around the clock, at some regular period. There's been a move towards using stimuli that are non-periodic and analyzing them with a different analysis protocol that's called population receptive field mapping. Who's heard of this? PRFs? Yeah, about half the people here. All right, this is a little bit complicated to explain, but I'm going to take my time, and if you don't understand it, I'll explain it again. So the idea is that every voxel is the sum of lots of neural responses that have receptive fields at some location in space. So you can model a voxel's response as a population of receptive fields. And the way that you do this is you have some model over here of what the receptive field of that population might look like. Here it's modeled as a Gaussian in the upper right quadrant. You then take your stimulus, which could be a rotating wedge, or it could be a bar, or it could be anything actually, and you multiply your stimulus by your model receptive field. Okay? You then take that and convolve it with a model hemodynamic response function. And this gives you a prediction of what the fMRI response might look like if the receptive field of your voxel is over here. Okay? You then compare this prediction to your measured fMRI time series and look at the residual. And if the fit is bad, you go back up here and you move your receptive field around. And each receptive field location results in a different prediction, and hopefully some receptive field size and location produces a prediction that matches your measurement. So this is a way of doing a kind of nonlinear search of all the potential receptive field properties that account for your data. And the big advantage of this is that you aren't constrained to using these periodically rotating wedges or rings. You have access to a much broader class of stimuli. And this might be more effective, or is thought to be more effective, at driving cortical areas in higher order regions, such as parietal cortex or ventral occipital cortex. Another advantage of this approach is that it allows a more full characterization of visual responses. So not only can you characterize where a population receptive field is, you get an estimate of its size, so you can measure the size of receptive fields. You can also use a richer set of models for modeling receptive field properties. So we know that V1 receptive fields actually don't look Gaussian. They have inhibitory surrounds that have a particular shape. They tend to be oriented. And there's a lot of computational modeling describing how V1 receptive fields are constructed from an array of receptive fields in LGN, and then beyond that from the retina. So the idea is that we can use that modeling information to derive an accurate model of V1 receptive fields and then fit that model using this fitting approach. There are lots of good tutorials online for how to do this. One of my favorite is from Dan Berman, who's a graduate student at Stanford, in Justin Gardner's lab. And this is a little Java applet for a web page. What you can do basically is draw a receptive field someplace and then rotate your stimulus through it. And that curve down here shows a predicted fMRI response. So the purple line is what the neurons are doing, and the light blue line is the model of what the fMRI response is doing. So as I rotate this wedge around, see there's no response. Maybe there's a little inhibition because neurons can be inhibited by activity outside the receptive field. And as I bring it around, all of a sudden there's a response. Okay, so this response here is going to be influenced by, for example, the size of the receptive field and its location. So a receptive field over here is always going to be driven by this stimulus. But if I pick a wedge, a ring, it's not going to be driven when the ring's out here, but it will be driven when the ring's over here. Okay. Similar for the traveling bar. So this is a nice little demo, I think. You can go home and kind of draw circles, make it go wild. And one of the, I think, most interesting observations using the PRF method is from Ed Silson, who's a postdoc in Chris Baker's lab. And he was one of the first people to show that when you use a non-Gaussian receptive field model and a PRF fit, you actually can see that in V2, a lot of the receptive fields are elliptical and point inwards towards the fovea. So this is a feature of neural responses that was missed by the standard retinotopic mapping protocol. So that's one of the major advances since the Courtney and Ungerleider review 20 years ago. Okay, so progress and challenges. What do we not yet know about retinotopic mapping? Well, the first thing is it takes a long time. We typically bring someone in the scanner for a full session to do retinotopy. So every subject in our experiments needs at least 2 sessions, one for retinotopy and one for the main experiment. And that's very inefficient. So one of the major lines of work is how to make this faster and more efficient. And there's a paper that's now under review by John Winower and Noah Benson that shows that you can combine noisy retinotopic mapping measurements, which are shown in the top row, with an atlas, which serves as your prior. So basically you fit an anatomical atlas to the data, combine it with some noisy measurements, and the combination of the two gives you a very accurate measurement of retinotopy. And you can actually do as well in 5 minutes of measurement as you can by combining these two, as you can from a whole session of retinotopy alone. So improving this, but the major caveat to this work, by the way, which is under review, and I'm wondering what the reviewers are going to say, is that this only works for V1, V2, and V3. If you care about higher order visual areas, which a lot of people do, you can't use this method. So one of the major challenges is going to be to extend this sort of approach to higher order cortical areas. I also want to say that you'll see a lot in the literature people using templates, either from the Human Connectome project or from that Wang atlas that I mentioned earlier. And those templates will produce pretty ROIs in lots of higher order areas, but they're incredibly inaccurate. So if you really want to target one of these higher order visual areas, you cannot rely on the atlas alone. You need some measurement from individual subjects. So that's certainly a challenge for the future. Another challenge is on the measurement side. So we are all painfully aware that we're not measuring neural activity directly. We're measuring the plumbing, and the plumbing is complicated. In particular, large draining veins terribly distort our measurements. And there's a really nice paper from John Winower, who was at Stanford at the time, showing that this big sinus here goes right through human V4 in the ventral surface of the brain. And that likely has totally misguided where people draw the boundary between V4 and V3. And also led to erroneous conclusions about the functional selectivity of V4. I think that's actually the tip of the iceberg. I think the veins have distorted our entire image of how the different cortical areas are organized. And we're going to need to rely on either methods of measuring and discounting the venous contribution, or measuring a signal that's not contaminated by veins at all, such as cerebral blood volume or vaso. But I don't know anyone who can do that effectively. I'm joking. Renzo, who's sitting over here, is the go-to man for this method. Okay. And lastly, an area for future development is clearly this. So for anyone who's done PRF mapping, this step over here, right, this nonlinear fit, where you're searching the space of receptive fields to maximize that, is really hard to do. There's a non-linear fitting routine in MATLAB, but it gets caught in local minima. We clearly need better methods of doing that. And the answer is probably going to be some Bayesian hierarchical model that searches the space more effectively. It also takes a long time, and there has to be ways of speeding it up. So I think those are the three main challenges for retina topic mapping. Okay, so I've covered the first topic from the Ungerleider review. Second topic is functional specialization. And in that review, they focused on four things: orientation, motion, color, and objects, faces, and letters. I'm not going to talk about color or objects or faces and letters. Those are huge topics and better left for another day. The Baker Lab in particular has done a lot on this last topic. So if you're interested, you should go talk to people in Chris Baker's lab. And of course, color is studied extensively by Bevel Conway in the NEI. So if you're interested in what we've learned about color in the human or monkey brain, talk to Bevel. But I do study orientation and motion, so I'm going to be telling you about some of my own data here. So we know that V1 neurons are orientation selective. This is work done by Hubel and Wiesel in the 60s, which won them the Nobel Prize. What this figure shows is that V1 neurons are strongly selective to stimulus orientation. So a vertical orientation doesn't drive this neuron at all, but a stimulus that's 45 degrees drives this neuron very strongly. So neurons are orientation selective. They have smooth orientation tuning functions. And across the surface of V1, there are orientation selective columns. And those have been characterized most successfully using optical imaging in monkeys. So this is an optical imaging result where each color indicates the preferred orientation selectivity of a pixel. Each column here is about 1 millimeter in width. Okay, so very small. So can we measure orientation selectivity with fMRI? This black grid here is a grid of voxels shown to scale. Orientation columns are much smaller than voxels, so you would think that this should be impossible. How could we ever measure this fine scale structure with big voxels? One solution is to just increase this resolution of the scanning. So you can use really small voxels, and this voxel size, which is 0.75 millimeters, should be small enough to measure this pattern of selectivity. And in fact, there's one group in the world that's been able to do that. This is the Minnesota group from Camille Ergabil's lab. And they've shown in 2 individual subjects that it's possible to measure patterns of orientation selective columns in humans. I should mention that there are a number of caveats to this study, and it's not been replicated by any lab or in any additional subjects from this lab since this paper was published about 10 years ago. So it's kind of an open question of whether this is possible in humans with fMRI. So a second approach that's been tried is to use sensitive machine learning algorithms to suss out orientation selectivity. And the logic behind this is that every fMRI voxel samples from a large number of orientation selective columns, but there may be subtle biases introduced by that sampling arrangement. So for example, this voxel here samples from lots of orientation selective columns, but there are a few more columns that prefer this orientation than prefer horizontal. And that summed activity might be slightly biased towards the green orientation. That might not be significant by itself, but if you use an algorithm that can pool these biases across voxels, you then should be able to pull out what orientation the subject saw. So specifically, this is a machine learning approach to decoding the orientation that was presented on a particular trial in the experiment. This method was covered very nicely by John Dylan Haynes and Geraint Rees in a review about a decade ago, showing that you can use support vector machines to draw decision boundaries that clearly differentiate between different classes of stimuli, even when individual voxels don't show selectivity by themselves. So this works for orientation. This has been demonstrated a number of times. And one of the projects that I did in my old lab at NYU was to test why this works. So what's the neural basis for orientation decoding with machine learning algorithms with fMRI? So to answer this, we return to our old retinotopic mapping protocol. This is the figure I showed you before where the stimulus slowly rotated around visual space. We adapted that method for looking at orientation selectivity. So down here, rather than changing the position of the stimulus, we changed the orientation of the grading. So subjects fixated at the center of the screen, and this oriented grading just slowly rotated around once every 24 seconds. It's a very boring experiment to participate in. I was a subject, as was my graduate student. Typically, we were scanned at 2 hours at a time, just fixating this grading. It's really not fun. And this is the retinotopic map that I showed you before, focusing on V1. This is the V1, V2 boundary. And the question is, what pattern of selectivity do we see when we look at the response to the oriented grading? This model here would predict that every voxel would get a different orientation preference, that the pattern would look like salt and pepper. Okay? And what we found was an orientation bias map, where there's a clear progression from blue to red to yellow to green and back to blue that indicates a progression in preferred orientation. So this is a coarse scale, a large scale map for orientation preference that matches the retinotopic map. And this had never been observed before. This was missed by decades of single unit physiology recording one neuron at a time. And the reason why, we think, is that this orientation bias is relatively small relative to the huge orientation selectivity that you see with single units. But because it's shared spatially amongst neurons in a voxel, when you pool neural responses in a voxel, you kind of amplify the shared response. So the columnar response kind of cancels out, but the shared coarse scale orientation bias is actually amplified. That's why we can see it with fMRI and we can't see it with single units. The bias is radial, so this is a pretty picture we created by taking each voxel's response in V1 and plotting it according to its position in the retinotopic map. So each line here is a different voxel. Its position on these two axes is determined by retinotopy, and the orientation of the line is determined by its preferred orientation preference. And you can see that all of the oriented lines point inward, and that's consistent with a radial bias for orientation. Excuse me. We then tested whether this map is what was driving orientation decoding, because there had already been this debate in the literature about whether decoding methods were capable of elucidating responses at the columnar level. And we wanted to test whether decoding orientation was due to this coarse scale bias, which we discovered, or whether it was due to decoding. So the way we tested that is we took a whole bunch of voxels from V1 and we grouped them according to their preference in retinotopy. So here are a bunch of voxels that are color-coded by their retinotopic polar angle preference. We then bin them by this preference and then average them to create these supervoxels. So these synthetic supervoxels are actually the average response of many voxels here. We then bin them according to different levels of granularity. So this is the average of about 6 voxels. Now this supervoxel here is the average of about 10 voxels, and so forth. We did that all the way out here so that we're averaging all the voxels together in V1 to create 2 supervoxels. We then tested decoding performance, and we found that you could decode even if you average everything into 2 supervoxels. So this coarse scale map was sufficient to support orientation decoding. We then tested random averaging, and if you average randomly, not according to polar angle preference, decoding drops to chance. So from this, we conclude that the coarse scale map is sufficient for decoding. We then did a complementary analysis where we basically removed the map. So remember I showed you 2 maps, the polar angle map and the orientation map, and they looked very similar. We subtracted one from the other. And then we asked whether the residual had any information about orientation. And when we did that, decoding performance dropped to chance. So from this, we conclude that the map is necessary for decoding. So this coarse scale map, which had not been observed in the physiology literature, is totally dominating fMRI measurements of orientation. So progress on this front since the Ungerleider review, I think, has been mixed. We're certainly trying to measure orientation selectivity in V1. We can do so with ultra-specialized high-resolution methods. But we've also discovered biases in orientation measurements that were not known before and that really complicate measurements. And I think the future is going to be kind of getting past those biases to get at more physiologically relevant measures of orientation. So what about motion? We know a lot about orientation and motion from decades of physiology. Can we measure motion selective responses in fMRI? So people who have been around NIH or really anywhere in the field have probably heard that there are two pathways to the visual system characterized by Ungerleider and Mishkin in the 80s. There's the ventral what pathway and the dorsal where pathway. And what I've kind of been describing here are responses in V1, which are at the beginning of that bifurcation. But the next step in the where pathway is area MT, which is motion selective. So can we measure motion selectivity in MT? Well, one of the earliest demonstrations was from Roger Tutel, and he showed that an area in the lateral surface of the brain, just ventral to the superior temporal sulcus, is strongly selective to visual motion. So if you contrast a flickering checkerboard, which strongly activates V1, to moving dots, you see strong activity in this lateral area here, and that's the human homologue of MT. So MT is motion selective. Turns out there's some rough topography in human MT, which, by the way, is very hard to measure in monkeys because in monkeys, going one neuron at a time, it's very hard to elucidate the entire retinotopic map. But in humans with fMRI, it's fairly easy. This is work from Huck and Heeger in 2002, showing that moving stimuli strongly activate a large complex on the lateral surface of the brain. This is the area that I showed you before, MT. However, MT is selective for contralateral stimuli, okay? So that's this area here, whereas MST responds strongly to ipsilateral stimuli. This is actually a subtraction of contra and ipsi. So MST has large receptive fields that are selective for the entire visual field, whereas MT is contralateral. So you can reliably identify MT and MST. This work has been followed up by people in Brian Wendell's lab, showing there's topography within MT and MST, which they rather annoyingly renamed TO1 and TO2. TO is temporal occipital 1, temporal occipital 2. I'm very ambivalent about which naming scheme to use. I prefer MT and MST. It's also been shown that area MT is direction selective using an adaptation protocol. So the way this experiment works, subjects view an adapter, which is dot motion in one direction. After they've fully adapted to that stimulus, they're then presented with a test stimulus that's either motion in the same direction or motion in the opposite direction. And if they've adapted to that motion adapter stimulus, the response to this test stimulus should be weak, right? They've adapted to this direction, there'll be a weak response to this test. But if they've adapted to this test in the opposite direction should show release from adaptation. There should be a large response. So the difference between adapted and opposite test is a measure of how direction selective MT is. And what you find is MT shows really strong adaptation relative to the other cortical visual areas, showing that MT is strongly direction selective. Okay, this is, by the way, something we've learned since the Ungerleider and Courtney review in '97. Okay, so we know that MT is direction selective, but can we measure patterns of orientation selectivity that we know to be there from the physiology? We know that MT neurons have direction selective columns, and within a voxel there should be columns that are selective for lots of directions. So I repeated the same experiment that I told you about earlier with orientation, but using a big motion stimulus that changed direction every TR. So subjects fixated at the center here, and they viewed this random dot motion stimulus again for 2 hours. Extremely painful experiment to do. And what we found, surprisingly, was a map for motion direction that was really strong in V1 and hardly present in MT or MST, which is kind of confusing, right? This flies in the face of classical views of what V1 and MT are doing. Now, V1 is motion selective, but there's no organization to it. There are no columns in V1. MT is strongly motion selective and has an orderly columnar architecture. So we expected to see more information about motion direction in MT than in V1. We then changed the shape of the aperture of the stimulus. So in the first experiment, we had a large stimulus aperture. That was the entire screen. In this follow-up experiment, we had 2 apertures, one in the left visual field and one in the right. And the motion direction was in opposite directions. And what we found is that within V1, there's a complete map of motion direction that seemed to follow the shape of the aperture. So you see this circle here that's blown up over here, and all directions are shown. So there's a map in V1 of motion direction that follows the aperture edge. And this is kind of a mystery as to why this is the case. This is another representation of the same data, by the way, pooled across subjects. Each dot here is an individual voxel from an individual subject. I think n equals 5 subjects. The size of the dot indicates the strength of the response, and the color of the dot indicates the preferred orientation. And the fact that you see a smooth progression in color around this circle indicates a map for motion direction that's influenced by the shape of the aperture. So one theory as to, oh, by the way, it's n equals 4, not n equals 5. One theory as to why we're seeing an effect of the aperture is there's some suppression along the path of motion. So in this stimulus, a dot appears here and then moves across the aperture and then disappears. And what we're seeing is the largest response where the dot appears, and the amplitude gets smaller and smaller as you move through the patch. Yes? Sure. Yeah. Or I can hear you from here. It's fine.
In this, in all these experiments,
Yeah.
they're not doing any task.
They are, actually. I didn't mention that. But in all of the experiments that I do, subjects do a demanding staircase task at fixation that adjusts its difficulty so that they're at psychophysical threshold. And any runs where their threshold goes out of some range, I exclude the data. I'm a true believer in doing tasks during stimulus experiments, and I actually think it's a really important point. The reason why is that when you do a task, you focus attention at fixation. There can't be any attentional confound. Okay? Attention itself activates a lot of the areas that I'm interested in. So I want to eliminate that. The other thing is, it's been shown that doing a task at fixation reduces the amplitude of stimulus-driven responses, which is bad, but it reduces the noise more, which is good. So it's a net gain. And the reason why it reduces the noise more is there are random fluctuations in the location of attention. And that itself drives activity, but you can't track those. You don't know where subjects are attending. So that unaccounted for attentional fluctuation is a source of noise in the data. So I don't want that. So even though stimulus-driven responses are smaller when subjects do a task, the noise is much, much smaller. So it's a net win. And by the way, I'm such a believer in doing tasks, I'm unsure how to interpret resting state fMRI, because in resting state fMRI, attention is all over the place. And I think those attentional fluctuations are a large part of what people are measuring. But that's a big tangent that I don't want to go down too much now. Okay, so subjects are doing a task. So the hypothesis is that this map is somehow related to neural suppression of the dot stimulus as it moves along its trajectory. And to test that hypothesis, we changed the aperture yet again. We had these 2 bands, rectangular bands of motion, and the dots could either move inward or outward. Okay, so there's inward motion for 20 seconds, followed by outward motion for 20 seconds. And what we found in V1, this is a very complicated figure. So this is cortical distance. The fovea is over here. The far periphery is over here. At the fovea, there is a preference for fovea outward motion. Okay? So a stronger response in this direction. In the periphery, there's a stronger response for fovea inward motion. And at intermediate locations, there's an intermediate response. So this is consistent with the notion that there's the strongest response when the dots appear, and the response gets suppressed the longer the dots stay on the screen. And our sort of toy cartoon model for this is that neurons in V1 are daisy-chained through a series of connections that involve lateral inhibition. And when one neuron is fired, he basically suppresses his neighbor. Then the dot enters that neuron's receptor field, and he's suppressed from neuron 1. He now does a better job of suppressing his neighbor, and so on. And as that dot moves through the chain of neurons, the suppression increases. And of course, that's very difficult to test with fMRI. We really need to test that physiologically. But interesting to note that this makes a very particular laminar prediction, because that suppression should be most evident in layers that have cortical-cortical connections. So superficial or deep layers, not layer 4, which receives primarily feed-forward drive from the thalamus. So this particular hypothesis would be perfect for testing with an fMRI method that can give you layers. No pitch, Renzo. Okay, so the take-home message from functional specialization is that fMRI has proven itself to be very good at measuring coarse-scale patterns of activity. What we really want are fine-scale structures, and we don't yet have the ability to reliably measure things like columns. But I think that's on the horizon. Yeah?
You say you are methods are performing a task.
Yes.
You're measuring this, and the tasks are their threshold, like they're
At threshold, it's like a physical threshold.
yeah, the difficult tasks, or
Hmm?
the tasks are a bit difficult, right?
Yeah.
So how do you mean to say that V1 doesn't play in learning of those? Doesn't learning affect your PAPs? Or will it not? Or how does that conform come in?
Right. So the tasks are at fixation. So if there is learning, I would expect it to be local in V1. There are fluctuations in the thresholds. I'm measuring psychophysical threshold continuously in the staircases. And I have regressed against these low fluctuations in threshold. And it turns out there's a global signal that might be related to arousal, not attention, but arousal, that play a role in things like luminance detection.
So you're saying which way do you go then? V1 is involved in learning or it's not?
I think V1 is, I think the effects of learning are evident in V1. I don't know if V1 is involved in learning. But I think the effects of learning, the luminance decrement task, are evident in V1. But the timing of the task is orthogonal to the timing of the experiment. So for example, this is a block alternation experiment where the direction of the dots changes every 20 seconds. The task has a jittered ISI and a periodicity of 3 to 4 seconds. So it's running orthogonal to the main stimulus experiment. And we've done dual tasks where we have subjects either do the task of fixation or the task in the periphery, and there's complete interference. They can't do both. So when they're attending to the task at the fixation, they can't do the peripheral task. Which makes me think that when I'm measuring here, is as close to what you'd get from a paralyzed monkey prep, where the monkeys are just given stimuli and they can't respond. I want to turn my humans into monkeys. Okay, so in the last few minutes, I want to turn to the last topic in the Ungerleider review, which is potentially the most interesting, but also kind of the most confounding. So there's been a lot of work on kind of bottom-up hierarchical processing in visual cortical areas and top-down influences on visual cortical areas. So by bottom-up hierarchical processing, I mean there's a chain of activity going from LGN to V1 to V2 and V3 to V4 and so on, right, down the visual area hierarchy. And as you move down the visual area hierarchy, or up, I guess I should say, patterns of selectivity change. But there aren't good models that explain that transformation of information from V1 forward. Most of the work has been in characterizing the nature of that selectivity. Look, we see phase selectivity in the fusiform base area, or we see place selectivity in the place area. But we don't know how we got that information from the feed-forward stimulus drive. And I think some of the most interesting work has to do with constraining the models of successive stages in the visual area hierarchy by the responses measured at the earliest visual stages. So one nice example of that, I went in the wrong direction, is some unpublished or soon to be published work from Tony Mofshin's lab at NYU, showing patterns of selectivity from V1 to V2 to V3. So in the first contrast over here, they compared naturalistic texture stimuli to visual noise. And it's known that that V1 is active equally to textures and noise. So when you do that contrast, you see no activity in V1 because V1, the two sets of images are matched for low-level stimulus properties such as spatial frequency and contrast, luminance. So V1 cares about those things. When you do the subtraction, V1 is not active. But they've shown in very nice modeling and monkey physiology experiments that V2 cares a lot about naturalistic textures. And V2, of course, feeds this information forward in the visual area hierarchy. So V2, V3, and so on are active by this contrast. They've then shown that lots of areas are active by visual forms. So forms are things like curved lines, faces, not just textures, but things that have shapes that might be relevant for identifying objects. And of course, forms activate lots of visual areas as well. But when you do a form versus texture contrast, you see that V2 and V3 subtract out. Okay, so these higher order visual areas are selective for forms. So you've got low-level visual features in V1, textures, naturalistic textures in V2 and V3, and forms beyond V3. And they've characterized that transformation using computational models that rely on banks of linear filters that are connected in a certain way that's supported by the anatomy. They've characterized this pattern selectivity with single neuron recordings in monkey V1, V2, and V3. And now they've measured a similar pattern in humans viewing the same stimuli. And I think the challenge is to fit models to the data that will predict this pattern selectivity based on responses in V1 and V2. And there's likely a laminar profile to that computation. So this is an area of work that's going to benefit a lot by having laminar resolved fMRI. A second example that I want to talk about is top-down influences on visual cortical areas. Now, this is such a huge area of cognitive neuroscience that I couldn't even begin to give a summary. All visual areas are modulated by attention, by expectation, by prediction, by arousal. So there are lots of sources of top-down modulation. But characterizing how those top-down signals interact with bottom-up incoming signals is really interesting. And I think there's a lot of room there for future experiments. One study that I've been involved in, was just published recently in cerebral cortex, attempted to characterize bottom-up and top-down interactions in a part of the lateral surface of the brain called the temporal parietal junction. This is an area that gets a lot of feed-forward drive from the dorsal stream, so it gets a lot of motion-selective information from MT and from V1. It also gets a lot of top-down information from parietal areas. So it's where bottom-up meets top-down. And in this paper, we discovered 3 new visual areas in the TPJ. The TPJ, or temporal parietal junction, is often called an area in cognitive neuroscience experiments. It's been attributed to all sorts of things, theory of mind. It's been implicated in autism. I don't know about those cognitive features. I do know that flashing a stimulus during passive fixation activates 3 separate subregions in the TPJ. Those areas are highly variable in location. So this is a histogram across 5 observers characterizing the percent overlap in those observers. So if you project every one of those observers into a common space, what you find is that there's basically no overlap. So if you were to do a Telluric average and blur your data, you'd think that it was just one area, or you might not see it at all. So there's a lot of variability in the location of the TPJ. but we can reliably identify all three areas in each individual subject. And many of these areas, or at least the posterior and central TPJ, are strongly modulated by attentional cues. So this is a bar graph that you probably can't see from the back of the room, comparing valid and invalid attentional cueing. This is a protocol where we have subjects fixate, and we say there's going to be a stimulus over here, pay attention to it, and then we present the stimulus over here. So when we see a larger response for invalid cueing, that means that this area is responsive to attentional cues. And we see really strong attentional modulations in the posterior and central TPJ. So this is a site that gets feed-forward stimulus drive, passive visual information activates this area, and it gets a lot of top-down information about attentional cues. And I think one of the challenges of the field is going to be to characterize how those two signals interact. And that's another area, I know I've been saying this a lot, but it's partially because Renzo's sitting in the front row here, that where I think just measuring responses in these areas is not going to be enough. We're going to have to characterize how these signals interact, and there are probably distinct laminar profiles for that interaction. So layer fMRI is the future, and that concludes my talk. Thank you. Okay, questions in the last 5 minutes? Did anything not make any sense? Happy to go back.
Can I see the slide before that for Twitter?
Yeah. This one, the outline, for Twitter. Future is layer fMRI. Thank you, Renzo. Everyone should read that Ungerleider review, by the way. I was really shocked when I went back and read it. It was literally kind of the reason why I went to grad school. And it's so relevant today. It's really a great paper. Yeah. Okay. Yeah, thanks. Sure.
Top down and bottom up, like you do it in electrophysiology, you would record and look at the time latencies to differentiate them more or less, or like look at the feedback and like look at the signal change after a stimulus or something. But with the fMRI spatial or like the time scale that you can measure from, you cannot differentiate this so easily. So how would you explicitly say it's a top down or a bottom up?
Right. So, this is a really big topic, and Peter asked me to talk about the future of fMRI. And I think laminar resolved fMRI is really in the future, but also a really important topic is multimodal imaging. So we're going to have to be combining our measurements with MEG or simultaneous EEG and TMS. To get that temporal information is going to be critical, but I think that's also not enough, because showing latency differences doesn't prove it's top-down or bottom-up.
Not just latency, like
Yeah.
the response profiles.
Sure.
That might tell you at least there's like feedback or fatigue, or you can argue it in a different ways. But with fMRI, you might be confounded with it. You cannot.
You can't.
So
It's just one piece of the puzzle.
So saying it is a top-down or a bottom-up, how sure are you? That's my question.
Well, for the bottom-up, I'm fairly confident because of the convergence of information from physiology and from computational modeling. What's really beautiful about this work, and I think everyone should go check out these papers, because it kind of sets the paradigm for the sort of work that I aspire to do. They start with a simple linear model of how V1 receptor fields are constructed, right? V1 receptor fields are constructed from banks of LGN receptor fields that are wired up in a particular way. Now, V2 receptor fields are then constructed from banks of V1 receptor fields. Okay, from that kind of anatomical constraints, we get properties of selectivity that you can then test psychophysically in humans or electrophysiologically in monkeys. Then that makes predictions about what sort of stimuli will yield to different patterns of selectivity, which you can then test with future fMRI experiments. So it's really an interplay of computation, physiology, and fMRI that I think is really powerful, and psychophysics. Yeah.
This is just kind of a more general related question. Do we know what the sort of the underlying structural differences in retinotopic organization? Are there like cytoarchitectural differences in neurons that have different orientation sensitivities, for example? Is that something we know, or are we still?
It's something we really want to know. Yeah, that's a great question. So there have been papers showing that PRF properties differ for different groups. So schizophrenic subjects have different PRF properties. So when you measure these PRFs, you get more information about receptor fields. Similar claims have been made about anatomy, and then linking the anatomical observations with the physiological observations is also really important. So if you can characterize things like cortical magnification in a group of subjects, do they have different anatomical substrates or connectional substrates? Measuring tissue properties, I think, is really important. That's so far outside of what I do, though I didn't feel comfortable talking about it. Yeah. That is relevant, by the way. I'll just scroll back to the work that Noah Benson's doing at NYU, fitting the atlases. So that's purely based on the folding pattern of the brain. So this is based purely on this atlas here. Is based on the free surfer registration across subjects, which is based on a few anatomical landmarks. But that can be improved, I think, by measuring tissue properties. So connectional, diffusion tensor imaging sort of things. Yeah. Okay. Thank you.